you didn't have to look at my messy office here. But for any of the students on the webinar, work-life balance is really important. And I actually took this picture in Patagonia a couple of years ago. And so that's how I find my balance, go out and do some hiking and things like that. So I actually just had my lab group in Colorado for Western Animal Sciences last weekend, and we went to Estes Park and did some hiking as a part of that. So, okay, so let's start with this slide here. Why do minerals matter? My lab is really interested in refining the roles of minerals, in particular trace minerals and vitamins in feedlot cattle health and performance. And that is because minerals are the little pieces that hold all the biochemical stuff together in the body. So you can't throw a dart at a biological pathway and not hit a mineral or vitamin cofactor that helps support those things in metabolism. So I've got some of those listed here on the slide, the things that we care about in cattle production, and then the mineral wheel here on the other side of the slide. And this is pretty complicated. I've got a colleague that likes to call this the cat's ball of yarn because we have all these interactions between minerals here. But I like to think of it as job security. We're never going to figure this all out. So we've got plenty of time for us and the next generation of students to help figure out how we could move towards more strategic supplementation of trace mineral to feedlot cattle. And so in particular, if we're looking here at mineral status on the bottom, and then whatever our performance indice is on the side, that could be health, it could be cattle, that are growth rates, carcass performance, we want to kind of shoot to be in the middle of this. And so really we're interested in how do we move away from these broad spectrum 30 parts per million of zinc for all beef cattle to something really specific that matches with what that cattle, those cattle are capable of in terms of performance, in terms of immune function, in terms of carcass quality. So I'm going to tell you two stories of zinc nutrition today. And luckily for me, I had two amazing PhDs who are defending in the next week. So they had lots of slides ready for me. So we're going to focus on two pieces in the feedlot. The first will be when cattle first are weaned and get trucked some distance. So we might have cattle who are raised on forage in your part of the country down in the Southeast United States, but you don't have a lot of feedlots in your neck of the woods. Those are in my part of the country here or over in places like Texas where it's nice and dry and those are good conditions for raising cattle. Often those cattle have to be on a truck anywhere from 8 to 20 hours, depending where they're being moved across the country to move to those resources. And we've been examining how transit stresses cattle and how that actually affects things like antioxidant capacity. And so we've been studying zinc as a potential way to mitigate things like muscle fatigue associated with transit stress. So we'll talk a little bit about that. And then the second part here, we'll talk about a story for how when we give anabolic implants to cattle and we increase their rates of growth, we are seeing an increased demand for zinc. So I'll start out by giving a shout out to one of my PhD students, Katie Heiderscheit, who is finishing, she'll be defending her PhD next Friday. And this was part of her dissertation work where we were really interested in examining what would happen if we looked at an organic zinc source, and we'll talk about the details in a second, on cattle if we gave them a chance to consume that zinc for at least 3 weeks or so before we put them on a long haul trucking event, and then looked at their lactate as a marker of muscle fatigue and looked at their performance after trucking. So Katie's is the first study to really tie some of our zinc work and transit stress work together. And so for this, she used a series of Angus steers who were from a single ranch in southern Iowa. The study was conducted last fall. We housed them in pens containing growth safe bunks. If you're not familiar with a growth safe bunk, essentially imagine a bunk that sits on a load cell. Only one calf can put his head in there at a time. He's got a little radio tag that sits in his ear. And every time he goes into that bunk, it says, oh, calf number 12 is here now. It weighs how much feed was in that bunk before the calf came in. And again, when he leaves, and it attributes that feed that disappeared during that visit by that calf to that animal. So now I can have, say, 6 animals in a pen, but each animal is my individual experimental unit from a statistical standpoint, because I know exactly what each calf consumed. So we had 3 pens per treatment, so 18 steers per treatment here, and we looked at 3 concentrations of zinc. So the first one is we added no supplemental zinc to our diet, and our diet actually more than met what the NRC would recommend for zinc requirements here. Our second treatment was based on the fact that there have been surveys done of industry nutritional consultants that basically suggest they completely ignore the NRC and feed whatever the heck they want to. It's hard to know how much of it is because they have private data that says that that's the right thing to do, or how much of it is let's just feed a little bit more for insurance sake. So that's one of the things my lab has been really interested in, is why are all of these guys feeding this very different concentration of minerals? And so we've been studying some of that. So for this, what we're calling our industry rate of supplementation, we added 70 parts per million of zinc from a organic bisglycinate zinc source. And then for our last treatment, what we call our super nutritional zinc, we added 120 parts per million of zinc from that same bisglycinate source. So this would equal about 150 parts per million total in the diet, because we've got about 30 or 40 in our control, and then added 120. This is based on some of our previous research where we have fed high concentrations of zinc along with beta agonists and seen an improvement in growth performance there. So basically looking as we linearly increase zinc, what happens to cattle performance? So just looking at this project timeline, these cattle came in and started on their zinc treatments. They received an anabolic implant to start promoting extra growth. And then 25 days later, we've got blood, body weights, and muscle samples. They were loaded on a semi-trailer, driven around the state of Iowa for 18 hours. They can never leave the state lines because I always forget to get health papers for these transit studies. And so legally they can't leave the state lines. So depending on the time of year, the drivers see a lot of corn or a lot of snow. So this was in December. Luckily there was no snow, but all the corn was harvested already. So the drive around the state of Iowa for 18 hours, this would be a typical kind of semi-loaded forest. Cattle came back, they're unloaded. We take blood, muscle biopsies and body weights. And then Katie took samples again 6 days later, and then at the end of this 28-day period. So this is really focused on when cattle first come into the feedlot. They've been on a truck for a long time. We know they're tired, they wanna lay down, they don't wanna eat. Could having more zinc in the diet potentially help them recover more quickly from transit stress, kind of bounce back, eat better, and get back up on feed and start performing for us, and ultimately be healthier. So moving right into results for this one. So the first thing we noticed is that we increase plasma zinc concentrations as we feed more zinc. So nothing surprising there, but this is actually just focused on the first essentially 6 days after the trucking event. So this would have been the blood sample before trucking, be immediately off truck, and then day 6 post trucking here. The red is our control steers. And the first thing I want to point out is that even our steers that did not receive supplemental zinc are all very adequate in zinc status based on plasma markers. Now, you can argue that plasma marker is probably a crap biomarker for things like zinc status, and I wouldn't argue with you, you're probably right. But these animals would have to be more like 0.8 to be kind of towards the deficiency, so they're all very adequate based on the information we have today. The interesting thing that we noted here is that when we took blood samples from these calves straight off the truck, so if you're not familiar with how cattle are trucked, they can't lay down during this time period. They actually stand up during that time, and that's safest for them, because if a couple lay down, they're likely to get stepped on by their compadres who are in the trailer with them. So they're actually standing up the whole time. And now think about when you're like holding a steering wheel and you're going down a gravel road or something, right? You get that little bit of vibration. That's what that steer is experiencing the entire time he is standing in that truck, right? He's kind of got that tenseness, that vibrations, right? So this is like a long haul exercise event for this steer. For 18 hours, he's standing up and he's basically experiencing some degree of muscle exercise, right? So we think we see this muscle fatigue when they get back off the truck. So in the human literature, they have noted that plasma zinc concentrations can increase immediately after exercise when looking at muscle. And then if you actually measure it more like several hours after exercise is completed, that plasma zinc is actually lower. So there's clear effects of exercise on plasma zinc, but I think we're still figuring out what exactly that mechanism looks like. From here, we basically notice that we get a larger concentration or a larger increase in plasma zinc for either of our supplemental zinc treatments, which sort of makes sense. And then they all kind of come down a little bit by the time we get to 6 days post-trucking. So we're really interested in this because this is a pretty marked increase in plasma zinc concentrations. And as I said before, these things don't tend to change very much, but it looks like we're getting distinctions between our treatments. So this is probably one of the most interesting pieces of data we have from this study. And this is actually looking at serum lactate. So serum lactate is routinely used as a biomarker of muscle fatigue. So when serum lactate goes up, it's an indicator that that muscle has had to move into kind of this fine line between aerobic and anaerobic metabolism. And it's starting to produce more lactate, which then the muscle excretes so that the pH doesn't drop too much in the muscle. It ends up in the serum. The liver picks it up and it can potentially go through the Cori cycle to produce glucose. That glucose goes back to the muscle to help keep that muscle in a positive energetic state. So I thought this was the best way to show these data, but let me set up this graph first. So again, our non-supplemented zinc are in control, are shown in red. Our industry, or that additional 70 parts per million of zinc, are kind of shown in the gold. And then the gray is our additional 120 parts per million of zinc. So these are the samples before trucking, statistically fairly similar. Controls are actually a little bit higher. This is the off-truck sample where we would expect muscle fatigue has occurred for 18 hours, right? These steers are having to stand on this truck. And what we can see, if we just look at these two bars here, we get this marked increase in serum lactate. It's very consistent with some of our other work if they're not zinc supplemented. But it is really striking to us, and we'll see how this ties into performance in just a second, that within those cattle receiving the 70 parts per million of zinc shown here in the gold bars, it's basically flat. It's actually numerically a slight decrease in their serum lactate. The reason this is really interesting is because there is a zinc-dependent enzyme called lactate dehydrogenase, and lactate dehydrogenase B, the isoform B, is responsible for converting lactate back to pyruvate, so then that could jump into gluconeogenesis pathways. So what we think is actually happening here, and would be supported from the more basic literature, is that these cattle who had better zinc supplementation, and therefore better zinc status, higher plasma zinc, as we just showed, were actually able to have lesser muscle fatigue in this case, because they actually are able to convert this lactate produced at the muscle level back to glucose more quickly. We're getting ready to do some PCR work from muscle samples taken from this study, so hopefully we'll be able to confirm, because lactate dehydrogenase B is one of the specific things we're going to look for. Our super nutritional zinc kind of end up, interestingly, being sort of intermediate, and that's kind of the story of this study. Our controls are always kind of the worst. Our industry level cattle kind of do the best. And then our super nutritional zinc is kind of intermediate. So we're not sure if that's just a little bit too much zinc, or we didn't get quite as much benefit as we did with the industry supplementation. Cattle recover from stress really quickly. They're super resilient animals, and we're back down to baseline here by our next sampling time point at day 6. One of the things we've seen in multiple studies now is if we have animals that have better trace mineral status before they go through a trucking event, or even if we give them something like a multi-min injection after trucking and help recover their status more quickly, we'll start to see those animals go back on feed more quickly and start to consume their diet more quickly. From a producer standpoint, you want your calf to get into the feed yard, have a drink, have a bite to eat, lay down and rest, but you don't want him to spend days laying down and resting, right? You want him to start eating so you can start performing. So this is really important. That actually, it's pretty neat to see. This is looking at percent of the dry matter intake before we trucked these cattle. So 100% would have equaled the same amount of feed they were consuming before we put them on the truck. So that would be our goal, right? To get back up to here. And essentially what this graph is showing you is that by day 2, both of our zinc treatments are back or slightly above what their feed intake rate was before trucking. And there's been some studies that suggest feed intake recovery can take weeks after a long haul trucking event. So it's pretty cool to see that this started to bounce back more quickly. Kind of got some variations here across the first 5 days. The student's also interested in feeding behavior. So she's additionally got camera data where she's measuring how much during this time these animals spend standing versus lying. So over our whole 28-day period after the trucking events, kind of our receiving period here, we see that feed intake is greater in our industry supplemented cattle. So those that we added the 70 parts per million of the zinc from bisglycinate zinc here. Our super nutritional are intermediate, and our controls are the lowest. So consistent with that faster feed intake recovery, we do see that when we fed these cattle more zinc before trucking and then continuing that after trucking, they want to eat more. Now, it's only good if they eat more groceries if they convert it into something for us. So this is looking at performance of these cattle, and really just going to focus down here on the average daily gain data. So this would be the first 6 days off of trucking, after trucking, This would be the next kind of 3 weeks, and this overall would be this very last one. So in this case, these are kilograms per day. So these are some stupendous gains, even once we get out for the whole period. We've got a linear increase due to zinc and an effect of no zinc versus zinc. And really what you can see here is that our control cattle have the poorest average daily gain or the poorest growth rates, even though these are still very good growth rates, as opposed to either of our treatments that get the supplemental zinc. So again, our industry is kind of our rock star here. There's something that we just hit this sort of a sweet spot in terms of where we had the best lactate response. We seem to have the best feed intake recovery. We seem to have the best intake and the best performance here. So in our future studies focusing on transit and zinc, we're really interested in what would happen if you didn't feed zinc before trucking, you could only feed it after. Maybe you were buying cattle from someplace that you had no control over the prior diets. We would like to focus in on this industry level supplementation and see what does that look like both before and after trucking. All right, so what have we learned so far in our zinc and transit work? When we supplemented steers with zinc, particularly at that extra 70 parts per million of zinc, we had better performance post-transit. They seemed to recover more quickly, and we had greater plasma zinc concentrations. This probably ties back to the improvement in the serum lactate that we saw in these animals, since it was really pretty flat in those industry level steers. And we think that that's a sign that we had lesser muscle fatigue in those animals. I wanted to make a comment here too. We have consistently seen in our studies that transit stress, especially these 8, 10, 16-hour trucking events, we will decrease antioxidant capacity as measured by things like the ferric reducing antioxidant potential by at least 10%. In little calves that we've trucked, we've dropped it by 30%. We're still trying to figure out what that means. But clearly trucking causes oxidative stress. And so we've been investigating things like injectable vitamin C or other ways to actually give them sort of an antioxidant boost before or after trucking to help them recover from a trucking event. So in this study, my suspicion is that our zinc benefit is related to muscle energetics and that lactate metabolism with lactate dehydrogenase B, and we're following up on that. But it could equally be a benefit of antioxidant status, as zinc certainly supports antioxidant capacity as well. All right, so I'm going to tell 2 stories here. So that was kind of my shorter one. All right, so if you have not been to our feedlot at Iowa State, I'll see if I can get my video to play here. Maybe not. Okay. So this is a drone shot of our barn. So our farm is just north of Ames. So it's really nice. We can have undergrads work for us and things like that because it's 10 minutes to campus. Our pens are set up as 6-head pens, and we have concrete bunks up here under roof. And then we're open to the south. We've got our own feed mill on site, although we're also building a very expensive, fancy new feed mill at Iowa State. So we're looking forward to being able to pellet lots of diets and do fun things with research there. And then this is our working facility that the students like to joke is in the next county because it's a long ways from pen 60 to that working facility. So we have 60 pens here in the barn. All right, the other piece of data that I wanted to share with you today was about our work related to anabolic implants. So both anabolic implants and beta agonists are growth promoting technologies that we utilize in the feedlot. An estimated 70 to 85% of cattle in the US and feed are receiving these different technologies. This is a gun that would inject pellets. I've got a picture of this on the next slide. It goes under the ear here, under the skin. And these pellets contain things like analogs for estrogen, testosterone, progesterone, or a combination of those things. So here in the US, we castrate bull calves for temperamental issues and also for meat quality issues. In a lot of other countries, they actually feed bulls in the feedlot. They won't actually castrate them. So especially for a steer, we're kind of giving him back some of the testosterone for the growth potential that we took away from him when we castrated him. And this is a really easy way to give a growth boost to these animals. These are very low dose concentrations. And actually statistically, when you look at meat concentrations from implanted animals versus non-implanted animals, they are very negligible in the differences, and they're fractional compared to things like the birth control pills and other things that people put in their bodies. So very limited impact on meat, but the return on investment, we add about 100 pounds of hot carcass weight to a steer just by using a single implant. So it's a huge return on investment. It keeps our cattle in the feedlot for a shorter period of time, which has benefits in using less natural resources, less impact on the environment. We get them out the door more quickly. So these are basically pelleted, or some of these are coated pellets and some of them are uncoated that are in this thing under the ear. And basically they slowly dissolve over time and release the hormone. So one of the things that we have noted is that implants tend to work relatively quickly. So a lot of them will work for about 100 days. But when we have looked at weights over that 100-day period, we see a lot of that performance benefit happens really early, like in the first 20 days. So we asked ourselves the question, this is a big growth improvement in these animals, and we know things like zinc are really important for growth processes like protein synthesis. Does this animal who's growing faster than, like when I implant him, I can increase his growth rate 25% overnight. How have we changed his biology, right? Like I haven't changed his diet or anything, so how are we supporting this extra growth? So we asked ourselves the question of what would happen when we start to look at zinc. So we started just by saying, let's look at cattle that we have not implanted or cattle that we have implanted. This is the average daily gain or the growth rate of these cattle in the first 14 days after getting an implant. And we get almost a 30% improvement in average daily gain. So a marked increase in growth rates just by implanting these cattle. What we subsequently saw is that when we looked at plasma zinc concentrations in these implanted cattle, we saw that we were dropping it. And we were very consistently, we measured this across multiple studies. When we implanted cattle, we were dropping plasma zinc by anywhere from 8 to 20% in the first couple of weeks after implant. It is really hard to move things like plasma and liver zinc. They just don't change very much. And yet here was some clear evidence that the body is perhaps seeking zinc to help support this rapid rate of growth. And so we see this precipitous drop in plasma zinc. And in this study, actually that persisted out through 73 days after implant. So this is our no implant, kind of stayed steady. When we implanted them, they dropped and then kind of held steady there. So this was some of our first evidence that said, hey, we better get in here and start looking at some different concentrations of zinc in these implanted cattle. So I'm going to talk about two studies here. First of all, let's start by talking about why we care about zinc. The running joke in my lab is that anybody who has a dissertation on zinc just wants to title it Zinc, It's in Everything, because it is. It's really funny, you have a PhD student right now who's studying both zinc and manganese, and they're like polar opposites. Manganese, it's involved in like 5 things. Zinc, it's literally involved in thousands of things, right? So from a research perspective, you come at it from very different perspectives. We're currently doing a lot of omics stuff with our zinc work, just trying to kind of look for biomarkers, looking for some of these changes. So metabolomics, proteomics, transcriptomics. Zinc is really important in a lot of things related to protein synthesis, as shown here. This is one of my favorite graphs. This is Oberlace and Prasad, some nice rat work from the late 60s. Essentially what they did is they had rats that they gave lots of different concentrations of protein in the diet, right? Go up to your protein needs, you should see a linear increase in growth, maybe better. What they found is that actually you could feed more protein, but you had to supplement zinc to these rats as well before you saw a growth response. So I've just highlighted a couple of them. So the rats that got 8% crude protein are shown here. When they did not get zinc, their growth rate was down here. When they did get zinc, their growth rate has more than doubled. So this would suggest that zinc might be a limiting factor when it comes to a protein response. Similarly, if they had the 20% crude protein, which would have been well above what these rats actually required, they actually see still a benefit by increasing the zinc. So fundamentally, our NRC, the Gospel of Nutrient Requirements, as I like to call it, suggests that the zinc requirement for all of stages of beef cattle production is 30 parts per million to prevent signs of deficiency. So that's whether it's a reproducing cow, it's a weaned calf, it's a stressed feedlot animal, whatever. And we think that we probably need to have some degree of separation in there. And so we're kind of trying to move towards some strategic supplementation of zinc. I already mentioned earlier that the consultant's survey says that they feed more like 100 parts per million of zinc versus the 30 that the NRC recommends. So this is some of the things we started with for where to study in these experiments. All right, so shout out to a wonderful PhD student, Elizabeth Messersmith. She did her master's with me on copper and implanted cattle, and now her PhD work has been with zinc and implanted cattle, and she defends her dissertation tomorrow morning. So I'm going to show you 2 of her studies here. And in this first one, we had 128 Angus steers, and these steers were in a 2x4 experimental design. So again, we're in the grow safe bunks. Animals are experimental units. Either half of the steers received no implant or half of them received a potent implant. This is going to be the highest concentration of TBA or testosterone analog that we have available, along with estradiol. Very generically, estradiol causes protein synthesis to increase, and testosterone causes protein degradation to decrease. They have some overlap. It's more difficult than that, or more complex than that, but that's a simple way to think about it. The net effect is protein accretion increases. So we then factorialize with an implant to receive no supplemental zinc, 30 parts per million, 100 parts per million, or 150 parts per million of supplemental zinc, all provided from zinc sulfate. These cattle were a little heavy when we got this trial started finally, and so they were only on feed for about 60 days. Just showing a diet composition if you're not familiar with what our typical feedlot diet. So here we'll just focus on the finishing diet fed at the end of this trial. Dry rolled corn in this part of the country, you'd have to be further south than here to get into things like steam flake corn for improvements in efficiency. Sweet bran is just a wet corn gluten feed that we feed here, and then dried distillers are a product of the ethanol industry. So lots of protein in here, as can be seen here based on our analysis, almost 14% crude protein, which I think is pretty spot on for our implanted cattle growth needs. And then the basal diet without zinc added was about 40 parts per million. So remember, NRC says 30, we're already above what the NRC would say is adequate just based on our basal zinc concentrations in the diet. And then we would have added the 100, the 150, et cetera. So experimental timeline on this one would have been implanted on day 0. Elizabeth took muscle biopsies on half of these steers on day 11. And then she has liver biopsies here at the end for status, and then body weight and blood along the way. All right, so looking at our trial results here. As I mentioned before, implants seem to have this effect, especially very early on, right, in the first 20 days. So on this trial, we really tried to dial in and focus on what was the weight gain, what's the performance, what's the effect on muscle, what's the liver zinc changes, what is happening in this animal in the first 20 days or so. So this is looking at rate of daily gain in the first 18 days after the trial started and they received an implant. So the first thing is there's an effective implant where average daily gain is increased about 9% in these steers, which actually would be a little bit lower than average, and might be because we were still on transition diets, not quite as much energy in the diet at that time. So this graph will be similar throughout the rest of the presentation. We've got our first 4 bars that are solid, did not receive implants. The next 4 bars that are kind of dashed or hashed here did receive implants. And then within this, we've got kind of this light color is the non-added zinc treatment, and then 30, 100, and 150 for kind of our brown, red, and gold respectively. Okay, so no effects of zinc really if we did not implant these steers, although numerically maybe a little bit of a zinc effect there. What we definitely see is a linear increase within the implanted cattle. And it really is that our zinc 150 treatment, or our highest treatment, are kind of our rock stars here in the first 18 days. So performance is markedly better in these steers when we gave them a higher amount of zinc. Now, I should backtrack a second and say, just remember, zinc and implants started on the same day. So in contrast to the transit study, where we fed those zinc treatments for about a month before we trucked them, right? Whether that zinc needs to get where it needs to be in the body, whether there's any local storage in the muscle, those are all things we remain to kind of investigate. In these studies, we started the zinc and the implant on the same day. So we recently published a review from Messer-Smith et al., this would be Elizabeth, from our lab, looking at the interaction between zinc and steroidal implants, or these anabolic implants. If you've ever gotten to play with BioRender, it's kind of a really fun software tool to build figures like this in. So Dathan made this for us. Essentially what this is showing is that one of the mechanisms through which estrogen or TBA work is through these membrane-bound transporters. So we've got things like the G protein coupled estrogen receptor, the androgen receptor, et cetera. And the one I wanted to focus here on is this epidermal growth factor receptor. So downstream of binding to these steroid hormone receptors at the plasma membrane, there is effects that result in things like epidermal growth factor and downstream signaling. And what Elizabeth found in this study was that when she took these muscle biopsies and looked at gene expression for some of the targeted players in this graph, I just showed one for today's sake. When she looked at those cattle that received 150 parts per million of zinc and were also implanted, she sees a marked increase in gene expression of this epidermal growth factor receptor. And in some basic work where they've gone in and silenced or ablated the effects of this epidermal growth factor receptor, you stop the anabolic implant effect on growth. And so this suggests that this is a really important piece of this mechanism, and perhaps suggests one of the mechanisms by which this high zinc treatment might have been positively affecting growth. This would suggest that we would potentially have more of this anabolic implant signal in these high zinc and implanted cattle. She's got about 30 other genes that she has, but for the sake of time today, I thought I'd just show that one piece. So Elizabeth also saw, concurrent with our other studies, or consistent with our other studies, that in this study, in about the first 18 days, when she implanted cattle, she sees about a 6% decrease in plasma zinc. And it is interesting that in some of these treatments that we fed higher zinc, the drop isn't quite as much. So again, I think if we can get the right amount of zinc at the right time, we're preventing some of that plasma zinc drop from occurring, and that's resulting in increased availability to things like the protein synthesis pathways that zinc supports. That's kind of shown here. This is plasma zinc across the different study times. So this is our full factorial, the no implanted treatments, the implanted treatments, the 4 levels of zinc, and then linear and quadratic contrast within no implant or within the implanted cattle. So we're really trying to ask, if you were implanted, what's the effect of zinc? If you were not implanted, what's the effect of zinc? Just a couple of things I wanted to point out here. We, of course, linearly increase plasma zinc regardless of implant status as we feed more zinc to these cattle. But it is interesting to note these plasma zinc concentrations do not get particularly high. So we're still in the 1.3 to 1.4 parts per million of plasma zinc. It's very adequate, but it's not into a pharmacological response. So one of the things that we often get questions about is, how do you know that this is a physiological response versus a pharmacological response? And I would say that this is some of the best evidence we have that we are not getting into an overspilling, bypassing the gut level protection of zinc absorption. These are still relatively physiologically normal. Just kind of wrap up this one by looking at some of the carcass data. So hot carcass weight, this is the dressed carcass. This is what a lot of producers would get paid on. And we basically see a tendency for zinc to linearly increase this within our implanted cattle. And that's really best shown kind of right here. So we go from about 376 kilos in our non-zinc supplemented implanted, which is still, bigger than our non-implanted cattle. And then as we increase zinc, we add carcass weight. And part of that is because we also improve dressing percentage. This statistically is really hard to pick up. You have to have a lot of power in these studies, but we keep seeing this in our zinc work, where as we feed a little bit more zinc, we increase the dressing percentage, which is the amount of the carcass that came from the live animal. This is the part that we can actually potentially use. And we think that this is saying that zinc is helping with that protein accretion and carcass accretion. So that could have a lot of implications, especially considering that, like, for this study, it probably costs less than $2 per head for the entire feeding period to feed this amount of zinc. So it's very affordable. We would more than make up that money. I think Elizabeth calculated the return on investment just based on hot carcass on this one is about $23. So that results in our effect here. I'll just show this to wrap up this study. We linearly improve overall growth in these animals based on that improvement in dressing percentage and improved hot carcass. No effects of increasing zinc when we did not implant these cattle. Again, I think that solidifies that we probably have a different zinc requirement depending on the growth rate. It's not as high when the growth rate is lower. It's higher when the growth rate is higher. So the idea would be, by the time we get around to the next NRC, can we say, I expect 2 kilos average daily gain, and therefore I can calculate what my zinc requirement for that animal really is, as opposed to potentially losing some performance because I didn't have quite as much zinc in the diet, or the opposite, I didn't need as much zinc in the diet. We want to consider both sides of that. So the second implant one that I'll use to kind of wrap up the implant story is, again, some more work from Elizabeth's dissertation. And this is her deputy, as I'm calling him right now, our new PhD student, Dathan, who is going to be carrying on with this work. We have a USDA funded project that he will start in January. And again, in this one, we wanted to look at implanted cattle and also beta agonist fed cattle. So we'll talk about that here. 92 Angus cross steers in this study, and this one was a little bit longer, some more typical terminal implant period of about 98 days, again, in growth safe. And so we have, I think she's got 18 or 24 animals per treatment here. These guys all got an implant, so we didn't have enough room in the barn to fully factorialize this with implants, so everyone had our potent implant. And then everyone went on the beta agonist ractopamine hydrochloride for the last 28 days of the study at the rate shown here. And then, so basically our point was, let's just use all the technologies. It should sort of maximize growth, and let's see what zinc looks like against that. So we fed 0, 100, 150, or 180 parts per million of zinc, again from zinc sulfate. So timeline for this study, she's got sampling, liver and others taken here on day 9 and 10. We've got muscle samples from this trial as well. After 70 days, the beta agonist starts, and the beta agonist was fed for the last month. So the beta agonist that we use, ractopamine hydrochloride, would be a beta-1 adrenergic agonist. And so this would be basically stimulating the G protein coupled receptor cascade. We get cyclic AMP, increase intracellularly, turns on things like protein kinase A, and downstream we get the net effect of an animal who increases lipolysis. They're mobilizing their fat stores, and they're increasing protein accretion. So essentially we're taking the NEFAs mobilized from the fat stores, from lipolysis, we're feeding the muscle, and we're supporting protein synthesis. So we only do this at the very end of the feeding period, and it's because cattle get fat exponentially quickly. It happens, it feels like it happens overnight, and it's expensive because you get discounts if they're too fat. So they get fat really quickly. So beta agonists help kind of shift that curve, so we can, instead of having those cattle get fat on us really quickly, they're actually continuing to put down lean muscle, which is then can be that healthy meat product that we can sell. So looking at the results of this final study, this was focused again very early in the feeding period. So this is average daily gain in the first 10 days. And what we see is a linear effect of zinc, and then our contrast of no zinc versus zinc. So remember, in our last study, we only went up to 150. So in our infinite wisdom, we were like, let's go higher. Let's see how high we can take this bad boy. So we added 180 in this trial. And basically what we see is that 100 is just as good as anything else. And that's going to kind of persist in this trial. But we see that when we gave these cattle zinc, regardless of the concentration, we have better performance than if we did not give them supplemental zinc. And again, our diet here would have well met NRC requirements. So we are limiting cattle performance if we don't add more zinc to the diet than what the NRC might currently recommend when we're using these growth promoting technologies. This is the whole pre-beta agonist period. So the first 70 days, again, we're fairly hot on the implant, but we're starting to kind of slow down a little bit. And again, we see this positive effect. Any of our supplemental zinc treatments do better than our non-supplemental zinc treatment. Okay, so this would be great. I should just end the story here because now it gets confusing. So let's talk about zinc first. Okay, so plasma zinc, this would be their starting status. And so this is actually accounted for as a covariate in this day 10 analysis. And again, this would be from going from day 0 to day 10, about an 8% decrease in plasma zinc, so pretty consistent with our previous work. And we have greater plasma zinc when we supplement really any of the supplemental zinc concentrations. Again, as much as I would bemoan plasma zinc as a good biomarker, really, except for in humans when they're super zinc deficient, that's about the only time it's relatively useful. There might be a case that we could make here that there's some sort of threshold for plasma zinc that might say we need this much plasma zinc to fully support growth. We might be able to find an optimal number in there. We're getting ready to do a pooled analysis of all the data we have so far and hoping we can maybe track that down. Okay, so this is where it all goes sideways. We get into the beta agonist period. And at this point, we pretty much have no effects of zinc on the first 10 days of the beta agonist period, kind of analogous to that first 10 days of implant. We typically see a lot of performance. They all gained better in the beta agonist period than they did before. So we know we get a growth response, but we don't see a zinc effect. If anything, we see this funky cubic effect. All grad students, you definitely want cubic effects in your stats because they are amazing to explain to people. So we kind of had this cubic effect going on where really our 150 are kind of our worst performers here. And it's pretty interesting. They have been our best in our previous studies. They're kind of our worst here. So again, we're starting to wonder if this is because the beta agonist doesn't put as much pressure on protein synthesis as that implant does. We like to think of beta agonists as having this huge impact on growth, but quantitatively, the implant actually has a bigger impact on growth. So we're wondering if maybe by the time we get to this point, everybody's got pretty decent zinc status, and maybe we're just not having much of an effect on the protein pathway here. We have muscle samples from both of these time periods, implants and beta agonists, and we're getting into dig into some, especially looking at things like phosphorylated AKT and markers of that mTOR pathway. Translating overall final performance here, we do actually see no overall effects of zinc on overall average daily gain or feed intake, but we do have this kind of cubic effect where actually our cattle fed the 100 parts per million of zinc are the most feed efficient. So this is how much gain, kilos of gain to kilos of feed. So you want this number to be bigger because it means that they gained more per amount of feed that they had to consume. So this in combination with some of our other data, like our hot carcass data, we really feel most comfortable from this study recommending that the 100 parts per million of zinc is probably some of the best way to capture performance. Again, our heart carcass weight is better when we fed really any of the concentrations of supplemental zinc, but numerically it's best here in the zinc 100 and not quite as much of a dressing percentage effect here. But again, I think these effects that we picked up early from the implant period. Interestingly, zinc has been shown by some people, like Dr. Spears and some of his work, to affect back fat thickness. And we actually linearly increase back fat in these guys, a numerical tendency, and that results in having a higher yield grade, which doesn't matter for the yield grade that these guys had, but you don't actually want higher yield grades most of the time because you get into fatness issues. All right, so just wrapping up here with liver zinc. We did actually, from within 10 days of zinc supplementation, linearly increase liver zinc. And within 10 days of beta agonist supplementation, we get this fun quadratic effect, which is really interesting. So both the 100 and the 180 increase liver zinc, but our 150 is actually not different from the control. And remember, this was the treatment that didn't do as well performance-wise during this beta agonist period. So it makes me wonder if something was going on with this treatment where they weren't able to support that growth for some reason. Okay, I think for the sake of time, I'm going to skip these next two slides, although it's a cool manganese story, and anybody who knows me knows manganese is one of my babies. So you can ask questions about it if you want. What have we learned? Increasing dietary zinc above what the NRC recommends appears to positively affect cattle growth, especially when we implant them. And we think 100 parts per million of supplemental zinc is probably where that needs to be. And that kind of fits back to our transit study as well. The last one is just a hint. I was going to talk a little bit about liver manganese and arginase, but I could talk about that if anybody asked a question. Want to acknowledge my awesome lab group. We were just in Colorado hiking last week together, and some of the funders and donators for products, Zoetis, Phytobiotics, and Elanco, and then all of the awesome people that have done all of the work presented here. I just get the joy of working with them. So with that, what questions can I answer for you?
Hi, so Dr. Hansen, I just wanted to thank you again for a wonderful presentation. For all of our guests at this time, feel free to unmute yourself to ask Dr. Hansen a question directly, or you could type them in the chat box and I can read them aloud. Here, there's some in chat I can start with. This is from Zara. Are there any further beneficial effects of zinc supplementation on lactating cattle / reproduction rate?
Yeah, that's a good question. There's actually some really cool work if you ever want to go Google something called Zinc Sparks. So Zinc Sparks is this neat work that's been done from Northwestern University, actually using bovine ovaries as a model, where essentially they have shown that zinc is really important in oocyte fertilization. And it's actually a part of the zona pellucida formation that happens to prevent things like polyspermy. I don't know exactly what those zinc concentrations need to be for supplementation, but we know zinc is very important in reproduction. It's also important in things like preventing high somatic cell counts, which could be important for dairy cattle production and lactation.
Wonderful. There's another one here. She says, thanks. There's another one here in chat says, from Savannah, when feeding excess zinc into the diet, did you find it affects other mineral metabolism and or cause unwanted side effects of meat quality?
Yeah, that's a great question. So we actually did a study a few years ago where we fed much higher concentrations of zinc, well over 200 parts per million, and we did linearly decrease tenderness of the meat product. And we think part of that reason is that we are increasing growth rates so quickly that we are not able to kind of keep up with some of the processes that affect tendernization. However, we actually just finished a USDA funded project looking at how zinc affects early postmortem metabolism. And we looked at muscle chemistry from the time that we harvest the animal to the time that the product is aged and then consumed. And essentially what we see is that some of these higher concentrations of zinc, like this 150 part per million, these muscle actually have lower pH more quickly, which results in turning on of some of the right processes that help to sort of chew up the muscle fiber, the calpains and things like that. So there's actually some more tender product happening more quickly, which could have implications for not having to age that product as long, which would help with storage issues at the plants.
Says thank you. And here's another question from Natalia. She says, do you think that increased zinc supplementation in cattle would transfer into increased zinc absorption in humans? She's thinking of growing children who would have an increased demand for zinc.
Yeah, that's another great question. And actually, zinc, iron, and the B vitamins are probably the three nutrients that we think of as being very high in our high quality beef product, right? They're a great way to get that into the human. And absolutely, we have seen in some of our samples where we have done like muscle biopsies after different amounts of zinc supplementation that we can change the zinc concentration in the muscle. So yes, theoretically, each bite full of steak from that animal should have more zinc in it as a way to potentially get it into, like you said, growing children, who are where a lot of the model research has come from, where actually you can have a zinc deficient child, supplement them zinc, and see an improvement in growth processes.
Stephanie,
If there's anyone else,
Jack, Jack Odell here. Can you hear me?
Hi, Jack.
Hi, Steph. It's very nice to see you. Welcome, and thank you for joining us and for providing a stimulating seminar here. I have to represent. I'm teaching my regulation of metabolism course this semester, so definitely shout out to you for covering a few pathways there that these students are saying. But I got to ask you a question about your observation on lactate and your comments on lactate dehydrogenase. I'm sure you'll remember from my course that lactate dehydrogenase is a near equilibrium reaction. So, it's really driven, the balance between lactate and pyruvate is driven by substrate, by NAD, NADH, more so than the amounts of the enzyme per se. You don't affect the equilibrium constant by increasing more of the enzyme, right? So I'm not sure I'm grasping your idea of how zinc changes in lactate dehydrogenase might affect your circulating lactate level.
Yeah, that's a good question. Boy, that made me feel like a first semester grad student again. So we actually think, unfortunately, in that study, we were kicking ourselves later. The sample that we're missing is the liver, right? We took muscle samples in those animals and we didn't take liver. And so one of the things that we want to look at in the future is not only what like lactate dehydrogenase A and B activity are looking at in the muscle, but also what it's looking like in the liver. In addition to, I'm trying to remember, Katie has talked to me about some of the enzymes. So I know in some of our metabolomics work and our proteomics work with transit stress cattle, we have seen changes in like pentose phosphate pathway and other things that would suggest that perhaps we're also changing the availability of the reducing equivalents to help support some of those reactions. Yeah. So I agree with you. I think we're just starting to dip our toes into some of the energetic stuff, and we don't claim to be experts in it. But I think it's super intriguing from a zinc metabolism standpoint.
Yeah, well, I just really appreciate you connecting in some metabolism here for the students that are they're squaring up for exam number 2. So probably a little bit of angst going on, and I see some heads nodding. So anyway, thank you for an interesting presentation there, Steph. Nice to see you.
Yeah, you too.
Hello. Thanks for talking to us about minerals. It takes you back to my graduate days in Jack Miller's lab, where he actually was the first to define the zinc requirement in dairy calves. So I'm wondering what's different in your measurements compared to the endpoints that the researchers that developed the NRC requirements are. Why didn't we discover this greater need earlier on and put that into the requirements?
Yeah, that's an awesome question. And Miller's sort of one of the people that is cited ad libitum, right? Like in a lot of our work. So awesome work that they did, and more kinetic work, right? And more basic work than even we could get away with today. I think hands down the cattle are different. So a good statistic would be between 1977 and 2007, a meta-analysis published by Capper et al. In 2012 would suggest that growth rates increased by almost 50% during that time. So I think if we think about proportionally, we'll have some increase in feed intake as a part of that. That's probably covering some of it, but we've just got such an improvement in the growth rates of cattle. Our hot carcass weights increase about 7 pounds a year over the last 20 to 30 years. So we're just producing a bigger animal every year, and we're doing a better job of getting more of that to stay on the carcass as lean protein. And to me, that's what the zinc demand is about. So for sure, if we looked at a different mineral like copper, I actually think that the industry consultants are feeding way too much copper, and often we could maybe even feed a little less than what the NRC recommends. So it's not going to be blanket increases. It's going to be more about strategic things. And I think it's the growth rate differences here.
Great. Yeah. So that sort of brings up a second question I had is how much of the extra zinc stays in the animal compared to going out in the feed, the manure, and you know, land spread and increasing heavy metals into the soils and things like that.
Sure. Yeah. And that tends to be a hot topic, especially if you look at places like the European Union, which have actually put limitations on how much zinc they can feed in diets. I think the important consideration there is that they are talking pharmacological concentrations, like pig diets that might have 3,000 parts per million of zinc, and we're talking about 150 parts per million of zinc. So a lot lower concentrations that we're talking about. We're much more in a physiological range. We will see absorption rates decrease a little bit when we feed some of these higher concentrations based on our balance trials, but typically will be between 16 and 25% absorption rates for zinc that we feed. Depends on the source too. And I think part of the reason why feeding this higher amount is important is because they don't seem to be very quick at changing that rate of absorption in our measurements, but you retain more, right? So 20% of 150 is more than 20% of 30. And so we're able to retain more in the body. So we definitely see that those cattle retain more zinc when we put them in our metabolism crates and look at things like urine output, fecal output, et cetera.
Thanks.
Mm-hmm.
So if there are any more questions, feel free to ask them. But if not, thank you all for your questions. Again, Dr. Hansen, thank you very much for a very stimulating presentation. This is a reminder that next week we'll be hearing from Dr. Hope Lima again at 12 p.m. And I hope you all have a wonderful week, wonderful weekend.
Thanks.
Thank you, Dr. Hansen. Dr. Goodell wanted me to let you know that she apologizes that her internet went out and she will be watching your seminar when I post the YouTube link.
Sounds good. You guys have a good day.